Hello, my name's Mrs. Tyler, and today we're doing a knowledge organiser looking at monoclonal antibodies. Now, this is only relevant if you do triple or separate science GCSE and you're doing the higher level. So it's not on combined or foundation, and it's not on the foundation level. So it's only triple higher. Because it's only triple higher, as that would suggest, it's a difficult topic. In fact, it's straight down from A level. So, as usual, I'm going to break it down as much as I can. I'm going to go slowly. You get to draw along. We're going to make something that looks like this, where we're going to look at how monoclonal antibodies are made. We're going to look at how they're used in tests such as pregnancy tests and COVID tests. And then we're going to look at other uses of monoclonal antibodies. So, as usual, you just need a piece of A4 paper, a landscape, and a pen. It's going to be a tricky one. Let's get started. All right, so monoclonal antibodies then is just higher triple content, like I said. So we only need to be doing this if you're in a higher triple group. So I'm going to put the title in the middle. And as always, drawing it in a cloud. I'm going to divide the page in half. And then this side is going to be divided into about a third from the bottom and 2/3 at the top. So I'm going to do 3 different sections today. The first section is going to be making monoclonal antibodies. The next section is going to be using it in testing for pregnancy and diseases. And lastly, we're going to be thinking about other uses. So there are 3 sections. We're going to start then with this section here, making monoclonal antibodies. Now, this used to be on A level. It's come down from A level, so it's quite tricky. The first thing I want to say is that monoclonal antibodies are when we get lots and lots of antibodies that will only bind to a certain antigen. So antibodies. Remember, an antigen just means a molecule on the outside of a cell or a substance that triggers an immune response. So something that's not normally inside you. And so we want to make lots and lots of antibodies that will bind to that. Antibodies, remember, are Y-shaped molecules, they're proteins, and these sites here are where it can bind on. Now, monoclonal antibodies are made inside your body every time you get attacked by a pathogen by B lymphocytes, a type of white blood cell. And so the only place we can get monoclonal antibodies easily is from B lymphocytes. But we don't use humans for that. Unfortunately, we use an animal, and it's often a small animal like a mouse. So here's my mouse. It's not a very happy mouse, unfortunately, because it's going to be injected with an antigen. So we're going to inject a mouse with whatever we want the antibody to bind to. So if we want the antibody to bind to a pregnancy hormone, then the poor mouse is going to be injected with a pregnancy hormone. If we want the antibody to bind to anabolic steroids, to test for steroids, then the mouse is going to be injected with anabolic steroids. If we want the antibody to bind to COVID virus particles, the mouse is going to be injected with COVID. Okay. When that mouse is injected with antibodies, it will have an immune response and it will make B cells. So we're going to inject a mouse with the antigen. Remember, the antigen is whatever we're looking for in this case. And then, unfortunately for the poor mouse, we're going to have to take some blood and we're going to have a look for a B lymphocyte. Now, the B lymphocyte that's made should be one that's going to make antibodies to whatever we've injected it, because we've just injected it. So we'll inject it, wait a couple of days, take some blood, and then we'll find a white blood cell. And we're going to hope that the B lymphocyte, the white blood cell, will make the antibody that's going to bind to that antigen. The problem is with B lymphocytes is that although the mouse will make quite a few for itself, it's not going to make enough to test the population of Britain. All right. So we need to be able to clone this cell, make millions, billions, trillions of them. And to do that, we actually join it with a cancer cell. So we get a cancer cell. Cancer cells grow, all right? They divide uncontrollably, mitosis, and that's a bad thing because it makes tumours, but in this case it's a good thing, because if we can get a cancer cell to make the B lymphocyte, then it's going to grow and grow and grow, and we can get lots and lots and lots of them. We can clone it, all right? So we combine the B lymphocyte with the cancer cell, and we end up with a cancer cell that can make antibodies. And we call it a hybridoma. And that's antibody is the same as that antibody there, and that antibody is going to bind to the antigen. And once we've got one of those, then we are going to clone them, which means we're just going to make lots and lots of them. We're going to put them and grow them so that we've got billions of them. We use huge sterile containers for these, full of liquid. I'm not going to fill it full of those, but they're all going to be making, cloning, dividing, making millions of hybridoma cells, all of them making this monoclonal antibody. And then we can just extract the antibody from the liquid, because it will be floating around in the liquid. I need to point out that it's really important that you know that all these cells are clones, which means they are all identical cells, which means they will make identical monoclonal antibodies. So once we've got millions, billions, trillions of identical monoclonal antibodies, all of them designed to bind on really specifically to a chemical, we can use them to look for those chemicals. For example, we can use it in a pregnancy test. So we're going to go first of all as if the person is pregnant, and then we're going to think about when they're not pregnant. So when someone is pregnant, they make a hormone called human chorionic gonadotrophin. You don't need to know that. Shorten it to HCG. And that hormone is going round in the woman's blood, but it will also be present in her urine. But it's present in incredibly low amounts in the urine. So we can't just do normal chemical tests. We need something really, really specific. We need monoclonal antibodies. So in a pregnancy test, there's a section where the urine is added. And if the woman is pregnant, that urine will have HCG in it. I'm going to just pretend that the hormone are those little triangles there. So if the woman is pregnant, she'll wee on the test strip, and some of the hormone will be in that wee, and it will also go onto the test strip. Next to the test strip, then, are a set of free monoclonal antibodies. Okay, so remember, monoclonal antibodies, they're proteins, they've got 2 binding sites, and those binding sites are specific to an antigen. These ones are specific to the urine. But on the other end, they've got a blue bead of dye attached. So these are free antibodies, which means they can move. So if the antigen is present in the urine, it will stick to these antibodies here. So I'm going to draw it on. So it will bind to these free monoclonal antibodies. And as the urine soaks through the paper and moves down through the test strip, those antibodies will move with it because they're not attached to anything, they are free. They then get to the first window. All right, so all of these types of tests have got windows on them. And in a pregnancy test, the first window has got antibodies stuck to the paper. All right, they've been chemically attached. And these antibodies are to the antigen, the HCG. But the antigen is bound to these antibodies with the bead. So as these antibodies flow past, these will grab hold of the antigen, which means they also grab hold of the antibody and the bead. It's crazy. Okay, it's a crazy complicated system. So what you actually get, these immobilized antibodies, are stuck down, and they attach to the free antibodies with the antigen, which means the bead attaches. So the free antibodies with the antigen attach to the immobilized ones, so does the bead. which means you get a blue line here because these immobilised antibodies are sticking the free antibodies to it via the antigen, which means all the dye also lines up. Now, if you put red beads on like we did with COVID tests and we were looking for the COVID antigen, then these antibodies would move down and they stick to the first line. The antibodies have got the red bead attached and they stay put, which is why you got that faint line starting to appear as these antibodies moved over and they were caught by the immobilised ones. So this is the test window. And in this case, she's pregnant because there's a line in it. The urine carries on flowing and it gets to a second window, which is a control window. And here we've got another set of antibodies, but this time they're not to the antigen, they're to this end of the antibody, the tail end and the dye end. So they catch the antibodies from the other end. It doesn't matter whether they've got an antigen attached or not, because they're not after that end, they're after the tail end. So there's another set of immobilised. And this is to the other end. So this equals the control. Because what we're doing here is we're testing to see if these free antibodies have actually moved down the test paper and made it to here. And if they have made it to here, then the test has worked, okay, because they've gone over the test antibodies. So this is the control. So all antibodies should stick here. So we get a second blue line. So in a pregnancy test, just like in a COVID test, you should get two lines if you're pregnant. You should get a line showing the presence of the hormone, this test, and you should get a line showing that the antibodies have moved. Two lines. If you're not pregnant, then there is no hormone, no HCG in the urine, which means that when you wee on it, there's just urea and water there. It means when it gets to this first set of antibodies with the dye on, there's no antigen, no HCG. Just like in this one, these antibodies are free, they can move. Free antibodies. And they move down the paper towards the test window. But because there's no antigen, they just flow straight past it. So there is no line. Because there is no antigen to attach. And then lastly, they flow onto the control one. And the control one, remember, is to these antibodies, but from the opposite end, the bum end, and it will catch those because they should have made it. So you do get that blue line. So that shows the test has worked. So if you're not pregnant, you only get one line. Really difficult. So we can use exactly the same thing, like I said, for COVID. Where the COVID antigen was present, it stuck to the mobile antibodies that stuck to the immobilized antibodies and made the first line. It then tested to see if the antibodies had made it and made the second line. If you didn't have COVID, there was no COVID antigen in your saliva, which meant it didn't stick to the free antibodies, which meant when they moved down with the saliva, it didn't stick to the first line, but it should stick to the second line because it's sticking to the other end, the dye end. Really, really tricky. Lastly then, because these antibodies are so specific and because they're so careful at what they attach to, we can use it to attach things to other things biologically. So we can use it, for example, to treat cancer. So here's our cancer cell. And cancer cells often have either more antigens than normal or weird cancer antigens. So there's often something different about a cancer cell on its surface membrane. So these are cancer antigens. Now, we want to kill cancer cells, but we don't want to kill healthy cells. So if we put cell-killing drugs in, toxic drugs, into somebody to kill cells, we're going to kill them. So we want to target those drugs so they only attach to cancer cells, and we can use monoclonal antibodies to that. If their binding sites are only complementary to cancer, then they will only attach the toxic drug to cancer, which means you should get a concentration of the toxic drug only near the cancer cells and not near the health cells. So it's a way of targeting drugs. So you attach a toxic drug to a monoclonal antibody, and then it will only attach to cancer cells. So these were originally called magic bullets, using monoclonal antibodies with toxic drugs attached, specific or only binding to cancer antigens, so they only stick to cancer cells. And a bit like in this test, we can use it to identify substances, not just in pregnancy tests or COVID tests, but we can use it to identify anything. For example, we could add it to a microscope slide with a sample on it, and we could squirt some monoclonal antibodies on to a substance, and then it will stick to the cells if the substance is there, and we can identify it. Monoclonal antibody with dye. So once again, we use ones with beads of dye on. So in this one, to identify substances. And this can be done in a blood sample, it could be done in a pot, it could be done on a slide. But the rule is basically to identify the substance, you add a monoclonal antibody that will bind to the substance with a dye on it. It will only stick to cells that have that substance. Sometimes you can just use the naked eye because you'll see a colour change. Sometimes you'll need to use a microscope, but you can see where the dye is. And therefore, you can see which cells have got that substance. And that's it. Really tricky. So your job now is to go away and learn this. And then I'd like you to keep an eye out for a test video. I'm going to make a short test video after this. It'll be in the link below. once you've had a go at learning this, so read, cover, recite, so covering it up, I'd like you to go and have a test. Well done.